So we come to the final talk before we'll have a little coffee break. So our next speaker is from the Animal and Plant Health Agency in the UK. We're talking about bringing fields into the fold, and specifically one of the aspects of this first session is integrating artificial intelligence with One Health. So to talk about that, please join me in welcoming Joan, Amaya, Cuesta.
Good morning, everyone. Thank you for having me today. It's going to share my screen. Second. I would like to give a thanks to the Infectious Diseases and One Health team for the opportunity to contribute to this amazing event. Artificial intelligence is the fourth industrial revolution. It will be fundamental to the One Health concept. It will become a critical tool in ending inequality and achieving global health. Today, I will share with you the work that we do at our organization, where we serve as a national reference laboratory. After introducing our work, I will guide you through the journey of the history of artificial intelligence from its early beginning to the present day. I will explain the basic concepts of artificial intelligence, making it simpler and clear. To then, I will provide an overview of how AI is being used today and discuss some key points that we need to consider when applying AI to the One Health approach. And finally, I will talk about my PhD research, which focuses on using artificial intelligence to combat rabies disease. The Animal and Plant Health Agency is an executive agency of the Department of the Environment, Food and Rural Affairs. We work to safeguard animal and plant health for the benefit of people, the environment, and the economy. The APHA Rabies and Viral Zoonosis team undertake research and surveillance on behalf of the United Kingdom government for rabies, hantaviruses, and SARS-CoV-2. We are the UK reference laboratory for both human and animal rabies. This is relatively unusual in the majority of countries, but our team includes staff from public and animal health backgrounds, and we work closely with our medical and veterinary authorities within the UK. The team undertakes statutory diagnosis for rabies, which includes all suspect cases in humans and animals, and also deaths in quarantine. We also provide surveillance for UK bats and serological testing for post-vaccination antibodies in human and pets. So let's begin with a roadmap to understand how we arrive at our current situation. Our journey starts in 1838 when Gerardus Johannes Mulder, a Dutch chemist, introduced the term protein to describe the nitrogen-rich substances found in all living organisms. Fast forward to 1902, when two German scientists made a scientific discovery. They independently proposed that proteins are composed of sequences of organic compounds known as amino acids. These amino acids fall into 3D shapes, forming the structure of proteins. In 1949, Frederick Sanger made the groundbreaking achievement by sequencing a protein for the first time. He identified the exact order of the amino acids in the insulin, confirming that proteins have specific and set compositions. This discovery was a major milestone in understanding how proteins function at the molecular level. Just a year later, in 1950, Alan Turing published Computing Machinery and Intelligence. In his work, he proposed the Turing test as a way to measure machine intelligence. At 1951, researchers at the University of Manchester made a significant leap by programming computers to play checkers and chess. And remarkably, the checkers program learned from its own mistakes, improving over time until it became skilled enough to compete with human players. This was an early example of a computer learning and adapting, an important step toward modern artificial intelligence. Then, in 1957, John Kindrew achieved another scientific breakthrough by mapping the first 3D structure of a protein. However, despite this success, determining the shapes of proteins remained a slow and a challenging process for the next 60 years. This highlighted the complexity of proteins and the difficulty of understanding their structures, even with advanced techniques. In 1969, Cyrus Leventhal made an important hypothesis. The order of amino acids in a protein determines its structures. This idea, known as Leventhal's paradox, highlighted the relationship between the protein sequence and its 3D shape. Then, moving into the 1980s, software developers began utilizing programs to screen libraries of chemical compounds predicted to bind specific targets. This approach, known as computer-aided drug design, revolutionized the way that we develop medicines. In 1994, the Critical Assessment of Protein Structure Prediction, CASP, was established. CASP is a series of experiments designed to assess the current state of the art in protein structure predictions. Its goals are to evaluate progress, identify advancements, and highlight where future research efforts should be directed. Researchers from around the world participate in CASP to test their algorithms against real-world protein structures. CASP has been crucial in advancing techniques for predicting protein structures, which are vital for understanding biology and disease. A major breakthrough occurred in 2020 when Google's AlphaFold won the 14 CASP competition by solving the problem of predicting how proteins fall. Then, in 2021, AlphaFold made another significant contribution by sharing the predicted structures of 20,000 proteins in the human body. Previously, determining a single 3D structure required years of research, substantial resources, and many effort from the PhDs. Now, with AlphaFold 2, predicting a protein structure can be done in a matter of minutes. These advancements represented a major scientific breakthrough in our time. After giving you a glimpse of what happened until AlphaFold, let's go into a bit more detail of what is artificial intelligence or AI. So AI is used to classify programs that mimic intelligence and human cognitive functions like problem solving or learning. Artificial intelligence, machine learning, neural networks, deep neural networks, computer vision, and natural language processing are related technologies, but there are slight differences between them. Machine learning is a big subset that contains everything. Neural networks, also called artificial neural networks, are a subset of machine learning and are the backbone of deep learning algorithms. They are called neural because they mimic how neurons in the brain signal one another. A neural network receives information in the input layer. It processes it through at least one hidden layer and then passes the result to the output layer and gives the prediction or the output desired. Deep learning or deep neural networks, on the other hand, is made up of several hidden layers of neural networks that perform complex operations. There are two branches of deep neural network programs, computer vision and natural language processing. Natural language processing makes it possible for computers to read text, sequences, and interpret their significance. Then computer vision enables systems to see, identify, and process images and videos. Artificial intelligence is starting to play a crucial role in advancing the One Health approach globally. AI algorithms are capable of analyzing vast amounts of data from human, animal, and environmental health. This helps to identify potential disease hotspots and predict outbreaks before they become severe. This early detection allows for timely interventions, helping to prevent the spread of diseases between species. Moreover, AI facilitates the integration of diverse data sources, which enhances our understanding of complex interactions between ecosystems. This is essential for developing causal models that guide public health responses and management strategies for zoonotic diseases. AI has the potential for real-time tracking of animal health and behavior. The monitoring and analysis of big data can quickly identify and flag concerns, allowing for prompt intervention in livestock, wildlife species, or suspected reservoirs of zoonotic diseases. Additionally, AI-powered analytics can process real-time data streams to detect outbreaks and evaluate their severity. This immediate analysis helps guide resource allocation and intervention strategies, resulting in a more effective and coordinated response to health crises. AI can significantly enhance the development of personalized treatment, leading to improved outcomes in both human and veterinary medicine. Using predictive modeling, artificial intelligence can optimize drug development and treatment protocols tailored to the specific needs of each patient or animal, ensuring more effective and targeted therapies. Moreover, AI tools will be instrumental in assessing and predicting the impacts of environmental changes on health outcomes. For instance, AI can analyze how ecosystems degradation may increase the risk of zoonotic diseases, helping to anticipate and address potential health threats before they escalate. One of the AI's most prominent applications today is in antimicrobial identification and the development of strategies to combat antimicrobial resistance. Researchers have identified new antimicrobial agents and optimized some existing treatments, improving our ability to manage resistant infections. Further to this, AI facilitates the integration and analysis of diverse data sources through unified platforms that combine human health, animal health, and environmental data. This interconnected approach promotes a more comprehensive understanding of health dynamics and supports more informed decision-making. The successful integration of AI into the One Health approach relies on several factors. For instance, developing comprehensive repositories that combine AI with traditional databases is essential. The seamless transition to digital records is still a challenge, and right now there is not an unified or standard approach to generate, store, and use the data. Effective implementation of AI will require strong collaboration among various stakeholders, including policymakers, researchers, and practitioners. Working together ensures that AI technologies are applied in ways that address the needs of all sectors involved in health, environment, and disease management. It is essential to train healthcare professionals and researchers in AI technologies so that they can use them effectively. Building capacity in AI skills will maximize its benefits in health management and research, ensuring that these technologies are applied appropriately and effectively. As AI technologies become integrated into health systems, public database, smartphones, and many other platforms, it is crucial to establish guidelines and oversight frameworks. This includes addressing issues related to data privacy, ensuring equity, and mitigating potential biases in AI algorithms. Responsible use of AI will ensure that it benefits all stakeholders and maintains the public trust. And most importantly, scientists and experts should be vital in establishing these guidance and rules. Almost every month, there are new publications of AI tools with accurate results. For instance, a self-supervised model trained on chest X-ray image achieved results comparable to expert radiologists. In another study, the model was trained with retinal image and was capable of predicting traits that human can never detect. Another example is a deep neural network that accurately predicted the host for some viral species. My PhD focuses on the application of artificial intelligence for the research of lyssavirus infection and analysis of lyssavirus-host interactions. To put you into context, I will explain about the uniqueness of the lyssaviruses. The Rhabdoviridae family consists of viruses with a single-strand negative sense RNA genome. They are characterized by their cylindrical or bullet-shaped form. Within the Rhabdoviridae family, we find the genus Lyssavirus with several species. I want to emphasize the distinction between the rabies virus and the disease rabies. The rabies virus or classical rabies virus is a species within the Lyssavirus genus, while the disease rabies refers to the manifestation of its characteristic symptoms and signs. In general, I will refer to rabies as a disease. The Lyssavirus genus includes several species related to the rabies disease, such as Mokola virus, European bat lyssavirus type 1 and 2, the classical rabies virus, among other lyssaviruses. Rabies disease is the perfect example of one health approach due to its intersection across human, animal, and environmental health. Rabies is a zoonotic disease. The primary carriers of rabies are domestic dogs, wild animals like foxes, raccoons, bats, and some livestock. This interspecies transmission highlights the need for an integrated approach that considers both human and animal health. Rabies is a preventable disease and is nearly always fatal once the symptoms appear, making it a critical public health concern. This necessitates coordination between public health authorities, veterinarians, and local communities to prevent and control outbreaks. Lyssaviruses are primarily associated with bats. The greatest diversity of Lyssavirus species is found in bats in Eurasia and Africa. However, the rabies virus has never been detected in any of the bats from Europe, Africa, or Australasia, but it is the only Lyssavirus species present in the Americas. Rabies virus is a multi-host virus infecting almost all mammals, while the other Lyssavirus species seems to be limited to restricted hosts or only one unique host in some cases. Rabies virus is usually maintained in hosts from the order Chiroptera, which is the animal order from bats, and the order Carnivora, the animal order of dogs, cats, wolves, and others. Rabies virus is particularly challenging to eliminate worldwide due to its unique ability to cross species barriers and establish itself in new hosts. These cross-species transmissions means that rabies virus can infect a wide range of animals, making it difficult to control and eradicate. And the virus capacity to adapt and establish infections in different species complicates the efforts to achieve the global elimination of rabies. The reasons behind rabies virus broad host range and the host restrictions seen in other lyssavirus species is one of the big questions in virology. The aims of my PhD are to develop an AI program to evaluate phenotypic traits from lyssavirus infected samples and to investigate genetic fingerprints in lyssavirus species to determine the host restriction patterns. Rabies disease has been well controlled in some regions, but remains a significant threat in others. So targeted interventions are essential. I aim to minimize the risk of rabies spreading to new areas or re-emerging regions where it was previously controlled. During an episodic such as a rabies outbreak in wildlife or an outbreak of another zoonotic virus, the AI tool will provide meaningful data to identify early warning signs of an outbreak, allowing for preventive action in human populations. And to finish, in September 28th is World Rabies Day. World Rabies Day aims to raise awareness and advocate for rabies elimination globally. It is an event designed to be inclusive, uniting people, organizations, and stakeholders across all sectors against rabies. In 2022, APHA launched the UK Bat Rabies Dashboard, an interactive platform on which we show the data of the bat surveillance program in the UK. And this year, on October the 1st, we will host a worldwide webinar for World Rabies Day. Speakers from Africa, the Americas, and Europe will explain how they control rabies in their countries and the current situation in their geographical areas. So if you want to know about how rabies is tackled, for instance, in Colombia, Nigeria, Brazil, Europe, or South Africa, please keep an eye of the Global Alliance for Rabies Control webpage where the event will be promoted. Thank you so much to everyone, and special thanks to Jessica Lavery for help and everything. Thank you so much.
Thank you, Joan. Do we have any questions from the room? Yep, over on the left.
Hi there. Thank you for your presentation. My name is Sten. I'm a student of the IDOHDAY Masters, and I was wondering whether you could reflect on what you say about an AI tool to predict potential outbreaks of new diseases. And I was wondering whether you could reflect on, for one, the enormous sample bias that we have to the more developed countries, right? And additionally to the pathogens that we already know cause some diseases. So is there a potential that we can develop a tool that could also predict, say, a zoonotic transfer of a disease that is currently not so clinically relevant, but when it does become zoonotic, then suddenly it does? Because I assume we don't sample these yet.
Thank you so much. This is a really good question. So there have been efforts to using genetic sequences and metadata from the environment to just try to make models to provide the prediction of, for instance, if like neglected diseases or any other viruses that we don't expect to jump into another species, could have any potential for outbreak or spillover. So there have been some trials onto that with just rotavirus, influenza A, and one with rabies virus as well. And as well, it's a matter of the data. The problem is like as well in Europe and in North America, they have like a massive influx of really good data, while now we are starting to just get more data from other countries, from Africa or South America, just to unify or just train those models onto that. So I think it's a matter of time that we can just start to increase the predictions capabilities and forecast even more different viruses. Because as well, not only we need the information from the virus, but we need information from the possible host. And if possible, other environmental traits that we can have, if we can have.
Any further questions from the room? No? Any online? No. I think we're all good. So if you could join me in thanking Dr. Amaya Cuesta. Yeah.
Thank you.
And with that, we'll be going to a short coffee break. If you could all be back in your seats for 11 a.m. We'll see you then.